First of all, as Laura said, I want to give thanks to the organizers for the invitation. So I'm quite excited with this presentation because this is the first time we conduct this kind of a study in the Balearic Islands. So first of all, a brief introduction of the situation in Spain. Every year in the news, we can watch cases of imported cases of Zika and chikungunya, and also autochthonous cases of dengue in Murcia and in Catalonia. So, which is the situation in the Balearic Islands? In the Balearic Islands, since the first detection of Aedes albopictus in 2012, the population is increasing. This is a figure of the results of a national surveillance in airports and ports. So we can see in the airport and the port the abundance of the Aedes albopictus is increasing. And also here on the right, we have the cases since 2009, the cases of these diseases transmitted by Aedes albopictus, chikungunya, dengue, and Zika, which are also increasing every year. So why happened this? Well, first of all, of course, the globalization. One people who is on an endemic area of dengue, Zika, or chikungunya can be infected with this virus and then can travel to another country. Maybe it's possible to the other the other side of the earth in a couple of hours. So so this is one of the main problems of this kind of autochthonous transmissions. And also the invasive species are increasing the range of expansion due to the climate change, the deforestation that leads to a creation of new breeding sites, and also the increase of temperature in higher latitudes that also increase the expansion of the invasive species. So well, this is a life cycle that you already know. These life cycles can be as complex as you want. So we have different habitats: rural, natural, peri-urban, urban, with different hosts, different species, and of course, these species of hosts and vectors can go from one habitat to the other habitat, and these vectors can act as a bridge vectors from one habitat to the other. So, as part of the postdoc project of Dr. Rafael Gutierrez and led by Dr. Miguel Ángel Miranda, we conduct this study in the Balearic Islands. For the people that don't know where are the Balearic Islands, are the islands on the east of Spain. The largest island is Mallorca, okay? So, and in the southwest part is the municipality of Palma. So we select 4 habitats, 3 urban parks, 3 peri-urban areas, which are the same we use for the AIM surveillance, and 3 rural areas, which are livestock farms, and 1 dog pound. So we conduct weekly collections from July to November. We set the traps in the afternoon and then we collect early in the morning. And we use these 3. We use 3 different traps in each side. BG Sentinel trap baited with BG lure, mini CDC with CO2, and light traps. So once in the laboratory, the sample were anesthetized, determined by a chill table, and then determined the cell score in order to be the level of engorged females. And then the engorged female will keep singly individually in Eppendorf tubes, and the other females were kept at -80 in a pools of at least a maximum number of 25 individuals. Then, as Laura said, I did a short-term scientific mission. Thanks to the IMCOST organization, it was a really nice stay for 1 month in the Bernard Notch Institute of Tropical Medicine in the arbovirology group led by Dr. Renke Luchten. It was very interesting because since my study of biology degree, I didn't. It was my last time that I perform a PCR and I pipetting. So because I'm a biologist, a field biologist, so it was a very nice experience to refresh all of this. So for the blood, with the blood engorged females, we extract the RNA and the DNA with commercial kits, and then we perform a PCR with different sensitivity. One with sensitive for mammals, and the other with for birds. Then with the non-engorged females, we extract the DNA and RNA with a robot. Okay. And the PCRs performed with the panflavivirus, planalphavirus. We also screened for avian malaria with nested PCR and Dirophilarias with qPCR. And then the amplification of PCR is sent to the NCBI GenBank. So these are the results obtained. The results were mainly the peri-urban areas are the site with the more diversity of mosquitoes. The most abundant mosquitoes are Culex pipiens, followed by Aedes albopictus. In total, we obtained 76 blood engorged females and 206 pools of non-engorged females. Then these results we can show the engorged females were mainly Culex pipiens. Maybe some of you know why we collect mainly the engorged females are mainly Culex pipiens, because I don't know that it's difficult to collect engorged Aedes species, but we have also Aedes species, don't worry. Here we can observe that in dog pound and in rural areas, the mosquitoes mainly bite on human and domestic birds and domestic mammals. Okay, if you go in more detail, here you can see the species. The most dominant species is Homo sapiens, human. In dog pounds, we have also domestic birds like turkeys, peacocks, chicken, pigs, because in this dog pound there are also abandoned animals that people use has at home as pets. That these I don't remember the name. These small pigs that people have as pets at home and then abandon, and the dog pound take this to the center. So, and also in the rural areas, because there are farms, so there are chicken and these other species. And it's quite interesting. The most interesting part is in peri-urban areas, there are a lot of wild birds that like robins and several species of birds. And in urban parks, cats and robin and Homo sapiens mainly. And in case of one Culiceta annulata engorged female we collect, was 100% Homo sapiens. The urban park Aedes albopictus Homo sapiens. And in peri-urban areas, the Aedes albopictus bites on cats and rats, and Aedes caspius in peri-urban areas in rabbits. So what about the pathogens?
1 minute.
Okay, sorry. The pathogens are mainly avian malaria parasites. Okay, so here we have the interesting thing is Dirophilarias repens, Aedes caspius, and then the non-engorged females we find also avian malaria, but an interesting case of West Nile virus in Aedes albopictus in your peri-urban area. Okay, so as bullet points or highlights, you can see that, Culex pipiens and Aedes albopictus are the most abundant species in these habitats. Mosquito fed upon the animals, the host they have in this habitat. Okay, so the Culex pipiens fed mainly in human, wild birds, in peri-urban areas, domestic birds, rural areas, in rural areas. Aedes albopictus fed on human, in dog pounds, and urban areas where these species, along with Aedes caspius, fed on wild animals in peri-urban. And well, malaria is present in the Balearic Islands, mainly in Culex pipiens species. Dirophilaria repens is it was isolated in other parts of Europe, but it is the first time in Spain that this is a species of Dirophilaria is isolated from Aedes caspius, which is quite interesting. And West Nile, there are studies of laboratory studies that show that Aedes albopictus is competent vector of West Nile, but here this is the first time in Spain. Even I'm not sure, but probably in Europe, that the West Nile is for the first time isolated from a field collected Aedes albopictus. I know that in USA, especially in New Jersey, they isolate West Nile virus from Aedes albopictus, but this is, from as far as I know, that this is the first time that we obtain West Nile from field collected albopictus in Spain. So I want to give thanks to my colleagues in the Bernard Institute, the AIMCOST, and the people who are connected and watching my presentation. Thank you very much.